Incredible breakthrough, a protein from tardigrades can protect human cells from radiation. Introduction. On February 26, 2025, groundbreaking research published in Nature Biomedical Engineering revealed a significant advancement in cancer treatment. A unique protein derived from tardigrades, commonly known as water bears, has the potential to protect human cells from radiation damage. This innovative development stems from collaboration among researchers from the Massachusetts Institute of Technology, MIT, Brigham and Women's Hospital, and the University of Iowa. The study focuses on a damage suppressor protein named Dsup, which allows tardigrades to withstand radiation levels up to 3,000 times greater than what humans can tolerate. Mechanism of action. The Dsup protein is instrumental in the survival of tardigrades under extreme conditions. It achieves this by binding directly to DNA, effectively reducing the incidence of radiation-induced strand breaks. This property positions Dsup as a promising candidate for protecting healthy tissues during radiation therapy, a treatment modality that approximately 60% of cancer patients in the U.S. undergo as part of their care. Research methodology. In a departure from traditional techniques that involve direct protein injection, the researchers employed messenger RNA mRNA encoding for Dsup delivered via specially designed polymer lipid nanoparticles. This method allows for temporary expression of the protein in targeted tissues, minimizing the risk of permanent genetic alterations. The approach was tested in mouse models where it resulted in a remarkable 50% reduction in DNA damage in tissues such as the oral cavity and rectum, regions commonly affected by radiation therapy. Potential applications. The implications of this research extend beyond cancer treatment. The protective capabilities of Dsup could be invaluable for astronauts exposed to cosmic radiation during prolonged space missions. Furthermore, the technique may be adapted to mitigate damage from chemotherapy drugs, enhancing the overall efficacy of cancer treatment. Future research directions. The research team plans to develop a human-compatible version of the Dsup protein to reduce potential immune responses. They also aim to enhance the efficiency of mRNA delivery and prolong its expression duration, which could pave the way for broader clinical applications. This research signifies a potential shift in how radiation therapy is approached, offering hope for improved quality of life for cancer patients and safer space travel for astronauts. Timeline of events. Preliminary findings. Over several years, researchers have studied the extraordinary resilience of tardigrades and their unique biological mechanisms. Study publication. The study detailing the use of Dsup to protect against radiation damage was published in Nature Biomedical Engineering on February 26, 2025. Successful trials. The nanoparticles were tested in models, demonstrating significant reductions in DNA damage while still effectively treating tumors. Source comparison. Various sources consistently emphasize the protective role of the Dsup protein and the innovative use of mRNA delivery systems. MIT News and Genetic Engineering and Biotechnology News focus on the scientific process and research implications, while the University of Iowa's report highlights clinical applications and future directions. Recent discoveries of additional Dsup-like proteins in new tardigrade species may further enhance the feasibility of this approach. Conclusion. The integration of tardigrade biology into human medical applications represents a pioneering step toward more effective and less harmful cancer therapies. The potential to transform radiation therapy through the use of Dsup could lead to improved patient outcomes and broaden the scope of protective treatments in various medical fields. As research progresses, the hope is that these innovative strategies will contribute to healthier cancer therapies, enable longer space missions, and provide real-world longevity treatments for future generations. The breakthrough involving the Dsup protein from tardigrades presents a fascinating intersection of biology, medicine, and technology, leading to potential advancements in cancer therapies and space missions. Here are some counterintuitive insights that emerge from the research and its implications. 1. Nature's resilience versus human vulnerability. tardigrades, often called water bears, can withstand radiation levels up to 3,000 times greater than humans. This stark contrast highlights the inherent biological vulnerabilities in human cells, particularly when subjected to radiation during cancer therapies. The counterintuitive aspect is that while humans often associate strength with size and complexity, these microscopic creatures demonstrate that survival mechanisms can be simple and efficient. The use of a single protein to mitigate damage reveals that less can sometimes be more in biological design. 2. Temporary genetic modification as a safety measure. The research employs messenger RNA mRNA to temporarily express the Dsup protein rather than directly injecting the protein itself. This approach, while innovative, challenges the common perception that genetic modifications must be permanent to be effective. By using mRNA, the researchers ensure that the protective effects are transient, reducing the risk of long-term genetic changes that could be harmful. This raises questions about the nature of genetic interventions and their implications. Temporary modifications could be a safer alternative in many medical scenarios. 3. Localized protection. The Dsup protein's protective effects are localized to the tissues where the mRNA is delivered, preventing the tumor cells from also being shielded from radiation. This specificity contradicts the general assumption that protective measures will uniformly affect all surrounding cells, including cancerous ones. It emphasizes the potential for targeted therapies that can enhance treatment efficacy while minimizing collateral damage, a significant shift in how protective strategies may be conceived. For broader applications beyond cancer, initially the focus is on cancer treatment, however. The potential applications of the Dsup protein extend to protecting astronauts from cosmic radiation and possibly mitigating damage from chemotherapy drugs. This suggests a paradigm shift where research aimed at niche applications like space health can yield solutions with widespread implications in everyday medical practices. The counterintuitive takeaway here is that advancements in one field, space exploration, could inadvertently lead to breakthroughs in another, oncology. 5. Biocompatibility challenges. The researchers plan to develop a human-compatible version of the Dsup protein to lessen immune responses. This requirement contradicts the traditional expectation that natural proteins would be inherently safe and well tolerated by the human immune system. This highlights a counterintuitive limitation of using naturally occurring biological materials in medical applications. What is beneficial in one organism may provoke adverse reactions in another. 6. Complexity and simplicity. The idea that a single protein can provide significant protection against radiation challenges the notion that complex solutions are always required for complex problems. This breakthrough suggests that sometimes nature's simple solutions can be more effective than intricate synthetic alternatives. It invites a reconsideration of how we approach problem solving in medicine and biotechnology. 7. Interdisciplinary research impact. The collaboration between disciplines such as mechanical engineering, biology, and medicine underscores how solutions to medical challenges often arise from unexpected intersections of fields. This challenges the traditional compartmentalization of scientific research, encouraging a more holistic and integrated approach to problem solving. 8. Public perception of genetic technologies. The use of mRNA technology, particularly in light of its recent prominence due to COVID-19 vaccines, may shift public perception of genetic engineering and biotechnology. The successful application of mRNA in this context could foster a more favorable view of genetic interventions, countering fears associated with permanent genetic modifications and potentially paving the way for broader acceptance of biotechnological solutions in medicine. In summary, the findings related to the Dsup protein from tardigrades not only present a promising avenue for protecting human cells from radiation damage but also challenge prevailing assumptions about biology, medical interventions, and the interconnectedness of different scientific fields. This breakthrough serves as a reminder of the complexity and simplicity inherent in nature and the innovative potential that lies in understanding and leveraging these biological mechanisms. The discovery of the tardigrade protein Dsup and its potential application for protecting human cells from radiation damage during cancer treatments is a landmark development in medical research. While the core findings of the study are being widely reported, several significant aspects are often overlooked. Yet they hold profound implications for future medical practices and research directions. Below are some of the less highlighted elements of the research and their significance. 1. Temporary expression through mRNA delivery. The use of messenger RNA mRNA to induce the temporary expression of the Dsup protein is a crucial innovation. This method contrasts sharply with traditional approaches that might involve direct protein injections or permanent genetic modifications. Most discussions focus on Tsup's protective capabilities, but the delivery mechanism itself is groundbreaking. Significance: using mRNA for transient protein expression minimizes long-term risks associated with genetic alterations. Given the concerns surrounding gene therapies and permanent DNA changes, this method could pave the way for safer applications in clinical settings. It could set a new standard for how protective proteins are administered, allowing for acute responses to environmental threats without altering the genetic landscape of the cells. 2. Localized protection without affecting tumors. The study emphasizes that the protective effects of Dsup are localized to the areas of injection, ensuring that tumor cells do not receive the same protection from radiation therapy. This aspect tends to receive less attention but is critical for the efficacy of cancer treatments. Significance: this targeted approach allows for the simultaneous protection of healthy tissues while maintaining the full potency of radiation therapy against tumors. It addresses a long-standing issue in oncology where treatment often leads to collateral damage to healthy cells, potentially leading to treatment discontinuation. The localized action of Dsup can enhance patient compliance with treatment regimens, thereby improving overall outcomes. 3. Potential for broader applications. While the primary focus is on cancer treatment, the implications of Tsup's protective capabilities extend far beyond oncology. The possibility of applying this technology to protect astronauts from cosmic radiation during long-duration space missions is an exciting avenue that is often overlooked. Significance: this application could revolutionize space travel, especially as missions to Mars and beyond become feasible. Protecting astronauts from radiation is a critical challenge, and Dsup could serve as a safeguard against the detrimental effects of prolonged exposure to space radiation. This highlights the interdisciplinary potential of biological research linking space exploration with advancements in human health. 4. Exploration of additional Dsup-like proteins. The research hints at the discovery of additional Dsup-like proteins in newly identified tardigrade species, which could enhance the protective strategies developed from this research. This aspect, while mentioned, does not receive the focus it warrants. Significance: the exploration of these additional proteins could lead to more effective or versatile radioprotective agents. If these proteins exhibit similar or enhanced capabilities, they could be integrated into the mRNA delivery system, broadening the therapeutic arsenal available for radiation protection in various medical and environmental contexts. 5. Addressing immune response concerns. The researchers plan to engineer a human-compatible version of the Dsup protein to minimize potential immune responses, a point that may not be fully appreciated. Significance: reducing the risk of immune reactions is crucial for the clinical application of any new therapeutic agent. By proactively addressing this issue, the researchers demonstrate a comprehensive understanding of the translational challenges that lie ahead. This foresight could expedite the pathway to human trials and eventual use in clinics, as immune compatibility is often a significant barrier in the development of biologic therapies. 6. Integration with other therapies. The potential for Dsup to also mitigate DNA damage caused by chemotherapy drugs is another crucial aspect that could be overshadowed by the focus on radiation therapy. Significance: if the Dsup mRNA approach can be adapted for use alongside chemotherapeutic agents, it could significantly reduce the side effects associated with these treatments, improving the quality of life for a vast number of cancer patients. This integrative approach could lead to a new paradigm in cancer treatment where protective therapies are administered in conjunction with traditional treatment modalities, enhancing overall efficacy and patient tolerance. 7. Implications for long-term longevity treatments. The overarching theme of longevity, particularly in the context of aging and cellular damage, is an important but often understated aspect of this research. Significance: as the population ages and the incidence of age-related diseases increases, the ability to protect cells from cumulative damage could have profound implications. The Dsup protein's mechanism of action could inspire new strategies for promoting healthy aging, potentially leading to therapies that extend not just lifespan but healthspan, the period of life spent in good health. Conclusion: the discovery of the Dsup protein and its application through mRNA delivery represents a significant step forward in medical science. However, the implications extend far beyond the immediate applications in cancer therapy. The potential for localized protection, reduced immune response, and broader applications in space travel and other therapies, along with the exploration of additional proteins, are critical elements that could redefine our approach to treatment and prevention in various fields. As research progresses, these overlooked aspects may very well become focal points for future studies and clinical applications, ultimately leading to enhanced patient outcomes and innovative therapeutic strategies. Here are 4 valuable points from the breakthrough research involving the Dsup protein from tardigrades, which can potentially protect human cells from radiation damage. 1. Mechanism of action and effectiveness. The Dsup protein damage suppressor protein from tardigrades binds directly to DNA, significantly reducing radiation-induced strand breaks. This mechanism is crucial, particularly for patients undergoing radiation therapy, as it allows healthy tissues surrounding tumors to be protected. The effectiveness of this approach is underscored by research showing that delivering mRNA encoding Dsup resulted in a 50% reduction in DNA damage in mouse models. This finding is particularly promising for cancer patients, as radiation therapy is a common treatment modality for approximately 60% of cancer patients in the U.S., many of whom suffer from debilitating side effects due to damage to healthy tissues. 2. Innovative research methodology. The researchers employed a novel method for delivering the Dsup protein to target tissues. Instead of using the protein directly, they utilized messenger RNA mRNA encapsulated in polymer lipid nanoparticles, which allows for temporary expression of the protein. This method minimizes the risk of permanent genetic alterations, a significant advantage over traditional gene therapy techniques. The targeted delivery approach was validated in mouse models, further demonstrating its potential for clinical application without compromising the effectiveness of tumor treatment. 3. Broader applications beyond oncology. The implications of the Dsup protein extend beyond cancer therapy. The protective capabilities of this protein may also benefit astronauts exposed to cosmic radiation during space missions, potentially enhancing human safety in space exploration. Additionally, the technique could be adapted to mitigate damage from chemotherapy drugs, expanding its application to a wider range of medical scenarios where DNA damage is a concern. This versatility showcases the potential of Dsup as a multifaceted therapeutic agent. 4. Future research directions and human compatibility. Looking ahead, the research team plans to develop a human-compatible version of the Dsup protein to reduce any immune responses. They aim to enhance the efficiency of mRNA delivery and the duration of protein expression, which could lead to broader clinical applications. The study highlights the importance of not only harnessing natural mechanisms for medical advancements but also ensuring that these advancements are safe and effective for human use. This forward-thinking approach emphasizes the potential transformation of radiation therapy and other treatments, ultimately leading to improved patient outcomes and quality of life. In summary, the discovery of the Dsup protein's protective qualities against radiation damage holds significant promise for revolutionizing cancer therapies, enhancing the safety of astronauts in space and potentially mitigating the side effects of other medical treatments. The ongoing research aimed at optimizing these applications is crucial for realizing the full potential of this breakthrough. Here are 3 famous historical cases that illustrate incredible breakthroughs in science and medicine akin to the recent findings regarding the Dsup protein from tardigrades and its potential applications in protecting human cells from radiation. 1. The discovery of penicillin 1928. Overview. In 1928, Alexander Fleming, a Scottish bacteriologist, discovered penicillin by accident when he noticed that a mold called Penicillium notatum had contaminated one of his petri dishes and was killing nearby bacteria. This serendipitous observation led to the development of the first true antibiotic, which revolutionized medicine and has saved countless lives since its introduction. Impact. Penicillin became widely used during World War II to treat bacterial infections in soldiers, significantly reducing mortality rates. The discovery spurred the development of more antibiotics, leading to the antibiotic era and fundamentally changing the landscape of medical treatment for infections. 2. The development of the polio vaccine 1955. Overview. In the early 20th century, polio was a significant public health concern, causing paralysis and death among thousands, especially children. Dr. Jonas Salk developed the first effective polio vaccine, which was introduced in 1955. Salk's vaccine used inactivated poliovirus to stimulate immunity without causing the disease. Impact. The introduction of the polio vaccine led to a dramatic decline in polio cases worldwide and has played a crucial role in efforts to eradicate the disease. The success of the vaccine demonstrated the potential for immunization to control infectious diseases and laid the groundwork for future vaccine development. 3. CRISPR-Cas9 gene editing technology 2012. Overview. The discovery of the CRISPR-Cas9 gene editing technology represents a significant breakthrough in genetic engineering. Researchers Jennifer Doudna and Emmanuel Charpentier developed this method based on a bacterial immune system which allows for precise editing of DNA in living organisms. Impact. CRISPR technology has transformed genetic research and biotechnology, enabling scientists to modify genes with unprecedented precision and ease. It has applications in medicine, agriculture, and biological research, including the potential for treating genetic disorders, enhancing crop resilience, and advancing our understanding of gene functions. These historical breakthroughs exemplify how innovative discoveries can lead to profound advancements in science and medicine, similar to the promising potential of the Dsup protein in protecting human cells from radiation damage. The recent breakthrough involving the tardigrade-derived protein Dsup presents a transformative potential for the future of cancer treatment, space exploration, and longevity therapies. As we look ahead to the next decade, several predictions can be made about the implications of this research. Healthier cancer therapies. 1. Enhanced radiation therapy. By harnessing the protective properties of Dsup, we anticipate a significant reduction in the side effects associated with radiation therapy. This could lead to improved treatment adherence as patients experience less pain and discomfort. Hospitals may begin to incorporate Dsup-based treatments into standard protocols, leading to a shift in how radiation therapy is administered. 2. Broader application in oncology. The use of Dsup could expand beyond traditional radiation therapy to include chemotherapy and targeted therapies. Researchers will likely explore its application in protecting healthy tissues from a wider range of DNA-damaging agents, potentially creating a new class of adjunctive therapies that enhance the efficacy of existing cancer treatments. 3. Personalized medicine. The ability to tailor Dsup delivery systems to individual patient needs may emerge, allowing for personalized cancer treatment approaches. As mRNA technology advances, we may see bespoke formulations that optimize therapeutic outcomes based on the patient's specific cancer type and treatment history. Longer space missions. For astronaut health and cosmic radiation, the use of Dsup to protect astronauts from cosmic radiation could revolutionize space travel, making longer missions to Mars and beyond feasible. By integrating Dsup into astronaut health protocols, space agencies may mitigate radiation-related health risks, thereby enhancing crew safety and mission success. 5. Development of space-ready formulations. As interest in space exploration grows, the formulation of Dsup-based protectants may be adapted for use in space environments. Research in this area could lead to the creation of portable mRNA delivery systems that astronauts can use during missions, ensuring continuous protection from radiation exposure. Real-world longevity treatments. 6. Aging and cellular protection. The protective mechanisms exhibited by Dsup could influence the broader field of aging research. As scientists delve deeper into the molecular pathways involved in DNA repair and cellular resilience, Dsup may inspire novel therapies aimed at extending healthy lifespan by protecting cells from age-related damage. 7. Innovations in gene therapy. The success of mRNA delivery systems in the context of Dsup could pave the way for advanced gene therapies targeting a range of diseases. This could lead to treatments that not only mitigate the effects of aging but also enhance overall cellular function and longevity. Future research directions. 8. Human-compatible Dsup variants. Future research will likely focus on engineering human-compatible versions of the Dsup protein. This could reduce immune responses and enhance the safety of Dsup-based therapies, making them more suitable for widespread clinical use. 9. Scalability and manufacturing. As demand for Dsup therapies grows, the biotechnology sector will need to develop scalable manufacturing processes. This could lead to collaborations between academic institutions and biotech companies, fostering innovations that streamline production and reduce costs. 10. Regulatory pathways in clinical trials. The advent of Dsup-based therapies will necessitate new regulatory frameworks that address the unique challenges posed by mRNA therapies. We may see expedited pathways for clinical trials, particularly for cancer treatments, given the urgent need for effective therapies that minimize side effects. Conclusion. The integration of tardigrade-derived Dsup protein into medical applications heralds a new era of cancer treatment, space exploration, and longevity research. As we forge ahead, the implications of this breakthrough could reshape not only oncology but also our understanding of cellular resilience and human health. By leveraging nature's own protective mechanisms, we stand on the cusp of remarkable advancements that promise healthier lives and extended exploration of our universe.